Hello.
Well, how are you? I'm doing all right.
I haven't managed to get back into my research at all.
So I found a good paper for, well, something you'd be interested in. It was about they were doing embryogenesis and COMSOL, and they were modeling
Oh,
it.
okay.
So we'll see how that looks, what that looks like. And then I don't know, we might.
If it isn't a tensegrity object, it won't. It'll just be an approximation.
Yeah, well, we'll see how they deal with it.
And then you make a model, and it is lasts over a longer, I guess, strain than other models, and it kind of matches what tissue does. It makes you kind of skeptical over other models that they don't last over the full range
Right.
of strain. So, oh well, we'll see. Does it do stress strain curves well?
I don't know. I didn't really look that deeply into the paper. I just
Or are they
got
just
it ready. Yeah,
they could be curve matching? That's
maybe.
COMSOL does curve matching really well.
Okay. Yeah.
Mm-hmm.
We'll start with this paper here, Organogenesis and COMSOL. So this is simulating organogenesis in COMSOL multiphysics, tissue patterning with directed cell migration. They've trademarked multiphysics, or COMSOL multiphysics, I guess. So you can't use that unless you're using COMSOL. So this is the abstract, and this is a recent preprint from the arXiv. It was put up last week, I guess. The abstract reads, we present a COMSOL multiphysics implementation of a continuum model for directed cell migration, a key mechanism underlying tissue self-organization and morphogenesis.
Yes, it's a continuum model.
Yeah.
Not a tensegrity model.
No, it isn't. The model is formulated as a partial integral differential equation, or PIDE, combining random motility with non-local density-dependent guidance cues to capture phenomena such as cell sorting and aggregation. Our framework supports simulations in 1, 2, and 3 dimensions with both zero flux and periodic boundary conditions. It can be reformulated in a Lagrangian setting to efficiently handle tissue growth and domain deformation. We demonstrate that COMSOL multiphysics enables a flexible and accessible implementation of PIDEs, providing a generalizable platform for studying collective cell behavior and pattern formation in complex biological contexts. So we have is this, we have a continuum model, okay, and they're looking at directed cell migration, and then they're looking at this PIDE model, which is a partial integral differential equation. And so this allows us to combine this sort of random motility, maybe a Brownian motion type movement with non-local density dependence guidance cues. So these are ways that you have random movement, but you also have guidance cues, and it's sort of a mixture of the two. And you can handle this kind of differential equation. And so then you can model things in up to 3 dimensions, and then you can reformulate that mathematically into a Lagrangian, so you can deal with deformation and tissue growth. Okay, so that's kind of the idea here. So in this article, we present a continuum model for directed cell migration, or DCM. This is where cells self-organize into complex tissues during development. And so it seems like they they have to do this trademark every time they mention COMSOL multiphysics, which is really kind of distracting. But so they've actually did another set of papers on this where they used COMSOL multiphysics to simulate biological self-organization through reaction diffusion mechanisms. So these are citations 2 through 8. The first one is simulating organogenesis in COMSOL. So there's a conference proceeding simulating organogenesis in COMSOL, deforming and interacting domains. That's another conference proceeding. This is all COMSOL conference. simulating organogenesis and COMSOL multiphysics with PDE models, um, simulating organogenesis and COMSOL cell-based signaling models. 6 is simulating organogenesis and COMSOL multiphysics image-based modeling. 7 is simulating organogenesis with COMSOL multiphysics software, phase field-based simulations, and embryonic lung branching, so that's an interesting paper. And then 8 is simulating organogenesis and COMSOL multiphysics, comparison of methods for simulating branching morphogenesis. So they, and then they have some other ones down here as well. So that's kind of where they start off from. So they have this body of work that they've been building, and this is one of those kind of, I guess this is an extension of that work. So, they've been able to simulate reaction diffusion mechanisms, tissue mechanics, and fluid flows. So all of those things, actually, okay, 9 and 10 then are tissue mechanics and fluid flow. We implemented these models on growing, deforming, or interacting domains using arbitrary Lagrangian Eulerian, which is ALE, or phase field methods. So, and represented individual cells with compartmentalized diffusion and signaling. So this is where you have these kind of of course, development is a growing, deforming, and interacting domain, all three. And they use these methods, arbitrary Lagrangian Eulerian, which is citation 3, and then phase field methods 7 and 8. So we talked about those references already. And so they represent the individual cells, but also with compartmentalized diffusion and signaling. So you have, a number of cells in a compartment, and then you link those compartments together. Moreover, we implemented efficient methods for parameter estimation and use image-derived geometries. Okay, so then I want to talk more about directed cell migration. It enables the formation of spatial patterns from initially homogeneous conditions, relying solely on interactions at the cellular scale. So that's just like the baseline condition, to pattern tissue on the much larger organ scale, which is a sort of elaborated condition. So you can actually, they can model things at multiple scales. Or this process occurs at multiple scales. DCM arises from the interplay between random cellular motility and guided movement, ultimately leading to phenomena such as cell sorting and aggregation. In biological systems, guidance cues for directed migration include gradients in chemical signals, which are chemo-taxis, substrate stiffness durotaxis, surface topography topotaxis, and differences in cell-cell adhesion or differential adhesion. So you have all these different things in biological systems that serve as these guidance cues. And this happens at the tissue level, where you have substrate, you have chemical gradients, you have some sort of topography, and you have, heterogeneity in cell-cell adhesion across like a population of cells or a sheet of cells. And so that's what they're kind of trying to get at with this biological phenomenon. They want to be able to model all of these things in a multi-physics setting. And so they're using numerical modeling. And so in their approach, DCM is described using partial integral differential equations, which are capable of incorporating both local diffusion and non-local interaction terms. And this gives us a more sophisticated dynamics. And so, of course, solving PIDs is pretty difficult. It usually requires custom-built solvers based on finite volume or finite element methods. So this is a lot of computational time. And so, you have to have something that's gives you the ability to handle all those computations. So that's where they kind of plug COMSOL multiphysics, and they have a toolkit for this. Furthermore, in tissue growth is an inherent aspect of developmental biology and morphogenesis. So, we want to be able to model that accurately. To handle domain expansion efficiently, we demonstrate how DCM models can be reformulated in a Lagrangian framework, enhancing computational performance while maintaining model fidelity. Okay, so they get into the sort of the mathematics here. So we introduce the governing equations of the PIDE-based continuum model. The model describes the temporal evolution of a cell density field, which is CXT, X being a vector, and T being time. So this is our cell density field here, and then this is our differential equation here, where we have a gradient, a directed gradient, and a directed J term, and it's the multiplication of that. And this J term is the cellular flux. So this J term is directional, and it's the cellular flux in one direction. Or it's directional, I guess. It doesn't have to be in one direction. In this case, it's consistent with the gradient. This flux can arise from a combination of random or pressure-driven motility and directed density-dependent migration. So basically, J vector is the direction of travel of the cells, and so they're all traveling kind of in the same direction or a similar direction, and it's kind of measuring maybe the mean heading of that, and then it's traveling down this gradient. Maybe it's the gradient, like a stiffness gradient or a chemical gradient. For a single population model, this leads to the following equation in n spatial dimensions. And so this is our J term here, J vector, and this is our term here, and this is where this is true. So here, capital D denotes the cellular diffusion coefficient, and small a represents strength of the directed motility forces. So you have this diffusion coefficient just describing the cellular diffusion, and then you have the strength of directed motility forces. How strong is that force? Okay, the model is designed to capture 4 key aspects of collective cell behavior. So the first is that cells exhibit motility, which can be diffusion-like or random or pressure-driven, thereby contributing to tissue fluidity. So you have these cells that are migrating. It's nominally random, although it can be directed by certain processes, sub-processes, and it contributes to the fluidity of tissue. So tissue is not solid, it's always moving around. Second, cells are able to sense their surroundings. They have sensory receptors, and they can sense these stimuli that lead to these taxic behaviors. So you have durotaxis and chemotaxis and even phototaxis, although I don't know how relevant that is here. This is represented by the integral I vector Cix, which integrates local densities within a sensing radius R, corresponding to a line in 1 dimension, a disk in 2 dimensions, or a ball in 3 dimensions. So the sensing radius is basically how far out from the cell it can sense things. And it basically integrates all of the density around it. So if the gradient is going from weak to strong, then it computes maybe like a line in front of it or behind it. And if it's moving towards it, or the line in front of it is moving towards the source of that gradient, then it's able to determine that part of the gradient, or that part of its motion, or that part of its sensory surroundings is the place where it needs to go, and so it moves in that direction. If you have a 2-dimensional disk, the same principle holds true. You go in the direction of the stronger signal, and that gives you directional movement. Or you have a ball in 3D, so it just basically works the same way, except you have movement in these 3 dimensions. So that's how that works. So each cell has a sensing radius R. It can sense this gradient that it's looking for. and it senses sort of the greater concentration of something and moves towards that. And remember, all the cells are doing this simultaneously. Third, cells interact and attract one another with a strength A within that radius. So they interact with each other as they all have the sensing radius. They can not only sense the some gradient, but also other cells. And so this is where they can, attract. They can exhibit other behaviors like that. So they can coordinate their behavior. And in this simulation, the attraction is assumed to be a spatial uniform phenomena, although in biology it would be spatially heterogeneous. It just makes it too hard to deal with when it's spatially heterogeneous. So in the spatial uniform case, we're dealing with something that's independent of distance within the sensing radius. So that's how you define spatial uniform. Finally, the domain is a finite carrying capacity, such that cells locally compete for space. So that means that they're all kind of competing for one space, and they're jostling for position. And they can only one cell can only occupy one place in space at a time. So if it's if there's an advantage to being in a certain point in space, or a certain advantage of migrating in a certain direction, the cells will crowd each other, and they'll undergo a jamming phase transition, where then they stop moving and they can't move anymore because they're too densely packed. And so that suggests that you have this mechanism for packing, this crowding effect, and it allows, but it also allows cells to not aggregate kind of just at random. So it kind of counteracts aggregation. There's this population pressure that moves the cells around, and it keeps them kind of moving around and not kind of getting stuck, although they can get stuck in certain cases. So in this model, the population pressure can be incorporated either into the random motility term, DC, the directed motility term, HC, or the sensing kernel, GC. So the sensing kernel is the volume and what they're sensing there. The motility term is the random movement, and the directed motility term is finding gradients to follow and things like that. And so in this paper, we focus on the special case where DC equals 1, HC equals C1 minus C, and GC equals 1. So they set them at these constants. And then so that's kind of explained kind of in another paper. And so then they do this non-dimensionalization. They non-dimensionalize the model by setting, where LC is the characteristic length scale. Here we choose LC equals R. So this is just kind of a constant value. The non-dimensionalized equation then reads, and this is the partial differential equation here, and then this is the other part here, where they give you, they vectorize it, I guess. Then there's domain growth. So to avoid computationally expensive remeshing during domain growth, so that means when the sphere or when the volume is expanding because it's growing, and they don't want to remesh the simulation, because remember, this is something that's approximating a finite element model. So they have to, kind of reset the mesh as it grows. You only have this, kind of predefined grid in a finite element model, and you decompose in some surface using that method. But of course, if the thing grows, then you have to remesh the thing. So that's what's happening here. So to avoid that, we transform the coordinate system from an Eulerian to a Lagrangian frame. So that's kind of where they come up with this idea of making it into Lagrangian. This enables a simulation of domain growth on a static geometry. We begin this transformation with the general form of equations 5 and 6, which we saw by incorporating advection by a velocity field, u vector induced by the expansion of the tissue, and then this is the equation for that. To transform equation 7, which is this, into from the Eulerian into the Lagrangian frame, we define the rescaled coordinate, which is here, where we have a stretching factor. We consider here uniform linear domain growth of the form this. The velocity field u vector can be expressed as this, and the gradient operator transforms as this. So we have these three different things: a gradient operator, a velocity field, and this rescaled coordinate system where we have a stretching factor. And so then we have a directional gradient L, which denotes the gradient Lagrangian coordinates, and that's with respect to this this growth factor here, this rescaling factor. Since u vector times the gradient, directional gradient, vanishes in the Lagrangian frame, and the divergence of the velocity becomes a velocity vector for uniform growth, and then dimensions equation 7, which is up here, transforms into this, number 12. The non-local interaction term, I vector L and Lagrangian, then reads this. Kind of walk through that. Okay, so that's the mathematics. And then this is the implementation in COMSOL.
I just wanted to say I thought Eulerian math was used because it was less computationally intensive, and you could actually do the same thing with Lagrangian equations.
Yeah.
That's sort of, so you don't necessarily have to mix and match them, but I suppose it's more computationally intensive if you use just Lagrangian math.
Yeah, I
Is
think.
Yes.
Yes. I think Eulerian is like the default, but they said that they needed to be able to build a model where the system grew or stretched out. So they're using the Lagrangian for that, and they don't want to use that like as the default because it's too expensive. But they have no choice because the Eulerian representation doesn't allow for it.
Okay. Yeah. I always thought the Eulerian math was a bit less accurate too.
Yeah.
Okay. All right.
Yeah.
I may have to study it.
Oh yeah.
Not happy about that, but anyway.
It takes a couple passes to get like what they're doing, like in terms of each symbol and everything. But we're gonna get in more into the trademarked COMSOL multiphysics here in the implementation. So all simulations were performed in COMSOL multiphysics version 6.3. Equations 5 and 12 from the last section were implemented as a coefficient form PDE. So they're using partial differential equations here. So this is figure 1. This is an illustration of boundary conditions for a two-dimensional domain. So remember we talked about how you can have 1, 2, and 3-dimensional sort of layers, or 1, 2, and 3-dimensional representations. This is the two-dimensional domain. This is where we have geometric coordinate mapping for periodic boundary conditions. So this simulation domain is omega. It's in the middle here. And then we have these things on the edge. So for the periodic boundary condition, we have general extrusion. We have then CMAP, which is outside of general extrusion. And then it's just expanding that simulation domain. And then for C equals 0, we have an expansion, a little bit of an expansion of the initial domain, but it's bounded by these Lagrangian terms here. And so that's how that works. So in the above implementation, it is necessary to ensure that the integral remains well defined near the boundary domain, where the integration region extends beyond the domain. This was achieved with the coordinate mapping, which we saw here, with these things on the outside edge of these squares. This was achieved with coordinate mapping. We illustrated the procedure here in 2 dimensions for a quadratic domain centered at 0, 0, 2-dimensional coordinates. The primary domain of side length L, which we saw, is embedded into a concentric auxiliary quadratic domain of side length L plus 2p. For periodic boundary conditions, the position for the outer square are then mapped to the inner square with the general extrusion operator using the following rules in 2D. So these are their rules here. So it's just kind of a stack where you have these different conditions under which the model operates. So you have this transformation from x to x plus L, and then it's this. If this is true, then we return something. We keep doing this for different combinations of x and y plus and minus L. For example, to implement the mapping y to y plus L 0 for this condition, which is the orange domain up in the figure, so it's this area here, general extrusion up at the top and C-map down at the bottom. That's our orange domain. One defines a new general extrusion operator. In this case, the 3 upper domains are selected with the source region ranging from, and then they say the xy coordinates of our source region. And then the destination map is then specified by setting the x expression to x, as we only want to shift the values in y direction, and the y expression to y plus is L, 0, which maps the selected region onto the orange domain labeled CMap, ranging from these coordinates in x, y. This procedure is then repeated for all 4 edges and all 4 corners, totaling in 8 mappings in 2D. For the 3D case, the squares are replaced by cubes and centered at 0, 0, 0. And then we have the Z component, which adds to the mapping in this form. So you're just doing the same thing with a new dimension, and you're getting this result. Okay, let's go to this figure here. It's figure 2. This is where we have a summary of the patterning space. So the cells are migrating around for a reason, and we want to see what the output is. So we can get these emergent cell density patterns in 1, 2, and 3-dimensional domains with periodic boundary conditions. So up here in A at the top, this is the 1-dimensional case where we have these bivariate relationships between X and C. And so we can see that there is a oscillation here for their different functions that they're plotting out here. If you go to 2D, you can understand what those oscillations are. They're basically these patterns. So you have these blobs that form in 2D, and they're aperiodic as far as I can tell. So they kind of cover, they have the same coverage as the 1D case. So if you think of these humps, these modes of the time series, or the distribution here, as these blobs at the top of these blobs, and that's kind of the same thing. It's just in 2 dimensions. And then in 3 dimensions, you get these blobs that are aperiodic with some variation along the edges, and you can see that that's kind of in all 3 dimensions. Okay, and then B, you have a variation of the initial concentration, C0, and migratory strength A. So you have variation of those 2 things. The migratory strength is on this axis. On the y-axis, the C0, which is the initial concentration, is on the x-axis. So they're varying concentration, and they're varying the strength of migration. So if you have a higher concentration and a higher migration strength, you can have a lot of motility. If you have a low migration strength and a low migratory strength, then you have very little movement, very little motility, and all the variations within. So we have 2 graphs here. We have where we have T20 and T1000. So they're generating this for different times. And you can see that when there is a low migratory strength, it's just a smear of a certain color. And then, as you start to get higher migratory strength, you get pattern formation at low initial concentrations, they're these blobs. At higher concentrations, they're these kind of stripes. And then at even higher concentrations, you have these inverse blobs, where the background in the lower concentration case becomes kind of the blobs here. So it's kind of interesting. It's kind of recapitulating what we might think of as chemical morphogenesis under those conditions. You see the same thing for running it for longer. You see, actually, for the low migratory states, you actually see some pattern formation where you get striping and then blob formation that's very ill defined. And then for these other cases, going down in terms of migratory strength, so greater migratory strength, you start getting these blobs, but they're bigger. And then you get these even more well-defined blobs with the greater migratory strength. So you can see how those two parameters interplay and contribute to pattern formation. What this suggests is that, of course, migration strength is key to pattern formation, but also concentrations, and we knew that from chemical morphogenesis, the concentrations are more, as important, but maybe not as important as we thought. It's interesting set of results. And so then they talk about zero flux boundary conditions. So zero flux boundary conditions presents a similar challenge to periodic boundaries, as the non-local integral must also be defined at the domain boundaries. Unlike periodic boundaries, however, this issue can easily be resolved by setting the solution in the added outer rim to zero, which ensures that the integral is evaluated consistently across the entire physical domain, including its edges. The flux is simply constrained by a zero flux Neumann condition, which is here. And so that's how they kind of fix this. It's the J vector that's fixed to zero. Then they have an initial condition, which is a spatial uniform concentration, C0, perturbed by random biological density fluctuations of amplitude, of a certain amplitude to break the symmetry. For simplicity, these fluctuations are sampled from a uniform distribution with a vanishing mean, so this is the term. And so they basically have this spatial uniform concentration that gets perturbed by these fluctuations that come from a probability density function that's defined here. So you have different amplitudes. It's roughly this variance of that distribution, and then that's what you have in the simulation that breaks up this uniformity. So again, we're trying to sort of describe these sorts of things mathematically, and I guess we get a reasonable result. Okay, so okay, go ahead.
I still don't see the stress strain curves.
Oh, well, I don't know if we're just getting into results.
Probably not going to get that.
We don't know. Yeah, we might not. Who knows? But yeah, we'll see. Let's see. So they have their default simulation parameters here. They kind of summarize them so we know kind of what they're using. This is an exemplary simulation on a large domain. Okay, that looks interesting. Yeah, they don't have very much in their results here. And then the conclusions are here. And let's see if we have any stress strain curves, because it's upon request. No, I don't think we do. It looks like we have some other figures here. Figure 4 is the impact of mesh element size and number of integration points. So that's here. This is where we have segmented spots for a mesh with maximum element size of 0.1 p. Scaling of spot area with mesh resolution, which is B, which is where we have these different element types. So we have quad types, triangular types, and with the max mesh element size divided by P, and the spot area on the y-axis. So the spot area goes up and then down, so it fluctuates as the mesh element size increases. And then C is impacted the number of integration points for the integral of the pattern. So again, we number of integration points, by the time we get to 5, we get the area settles down. So the area kind of fluctuates as the number of integration points sort of gets established. And then the number of spots remains constant at 9. So our average spot area is 2, our number of spots is 9, and this is like, after we get to 5 integration points, we can get higher, and the model converges upon that. So that's kind of what they're doing here. And then the impact of growth speed on patterning. So the lesson here is that slower growth, which is represented in A, leads to fewer, more spaced out spots than faster growth, which is in B, on domains of the same size. So if we have slower growth, we get spots that are fewer in number and they're more spaced out. Faster growth leads to, more, I guess, a greater number of spots that are less spaced out. So it's like it's growing fast, it's producing more spots, and it's more dense. So that's kind of the lesson there. Okay, so that's, I think that's it for this paper. I'm sorry I didn't give you a stress strain curve. They do go through a lot of the stuff that they, their methods, and I think the point of this paper was to kind of, they have this cottage industry of COMSOL multiphysics papers where they're modeling different aspects of morphogenesis and embryogenesis. So they might have one in one of those other papers. I don't know.
They are kind of going off on a tangent in the wrong direction, is my opinion.
You think?
It's interesting, though. It's interesting research and might apply to other things like batteries or something. But as to physical cell growth, not as much, although if they included the physical hardness of something in that model, they might get somewhere with that.
Yeah.
Not sure. Yeah. You need to improve the elasticity of cells.
Yeah. And then they kind of mentioned that, and they said it's a possibility, but we're going to show kind of like pattern formation just as a function of some of these parameters, which is, fine, but it's like pattern formation.
Yeah.
It's, putting the cell in context, or if you have pattern formation, like if the stiffness is different of the substrate, what does that look like?
Yeah, and stiffness is instead of chemical gradient, if you go into stiffness gradient, and they did have some of that in there, I believe.
Yeah, they did.
Stiffness gradient, it moves faster than diffusion. Like the stiffness can change rapidly, whereas chemical diffusion is a slower process.
Yeah, I think that's important to point out, is that not all gradients are equal in terms of how the response of what's going on. So
response time.
Yeah,
they're all important, and diffusion is important, of course, but it has a different time scale, which is so you have to watch the time scale of some of these things.
Yeah.
Anyway, I don't know.
Well, it was a lot of math, and I'm really I'm always fascinated by the different mathematical models and the assumptions they make, and sort of the ways that they employ different techniques. So like we're learning Lagrangian mapping. We're using Lagrangian methods for growth, but you don't want to use that all the time because it's computationally expensive.
So it's interesting, but
yeah. Now, I've not used COMSOL that much for embryogenesis, so I don't really know if how well this is done. I should go through one of the CopyCell 3D papers and go through maybe as a comparison point,
Yeah,
Big, yeah,
CompuCell 3D does what they were doing there. It's does chemical gradients and physical gradients. Yeah, CompuCell 3D should be it, you should be able to do that with it because yeah.
I mean, yeah. I guess the point there would just be to compare the methods of how they arrive at their results, and if that
Yeah,
yeah,
see if they're using the same type of equations. See if you can replicate the results with each platform. Would be interesting.
right.
I think I'm going to try to stick with tensegrity for the paper I'm going to write, and I don't know if I want to get into the continuum mechanics too much. Although I'm supposed to do a literature review, but that's how a leopard gets its spots, and I don't know how that relates to the stress strain curve of a physical object. Anyways, yeah, it's too far out there, so it can look like it should.
Now, what does the literature look like on the continuum mechanics of tensegrities?
Yeah, continuum mechanics of tensegrities. That's an idea. I'll look that up as a broad term to find some more papers. I don't know if there is a continuum mechanics of tensegrity.
Oh, okay, so that's not necessarily a thing. It's just kind of.
Well, people have to do that to get some of their models to work in some of the commercial programs.
Okay.
So
they have to do a workaround, and then see how it performs.
Yeah, because it's not like tensegrities will all of a sudden change shape, and then the whatever commercial package you've got says, the building just fell down.
Yeah.
Well, it didn't. It was a tensegrity, and it just changed shape.
Yeah.
That's all. Didn't fall down. The microtubules just gave way in part of the cell, and it just changed shape, and it's still alive and still functioning the way it should.
Yeah.
Mm-hmm.
Very good.
I don't know.
Yeah.
Yeah.
All right, that sounds great. Thanks. So that's that paper. Hope that was useful in some way. I see Morgan's here. Hello, Morgan.
Yes, I just still just dropping kids here.
All right. Well, good to see you here. Let's see. I wanted to do something else here. Like get into this paper, and this is going to be. I'm not going to get very deeply into it, but I wanted to talk a little bit about it, just as a, this is a new arXiv paper from 2025, from I think a couple weeks ago, July 31st. So this is from Biological Physics. That heading subject category. So this is Arvind Murugan and colleagues, and this is called "Could Living Cells Use Phase Transitions to Process Information?" So this is, heavy duty biophysics, and considering phase transitions as sort of a source of information processing. So we talked about jamming phase transitions in the last paper, that possibility that cells can, as they're migrating, if they get to a certain density, can stop migrating because they get stuck or jammed together. And so then that can be like a way to sort of limit diffusion, or it can initiate fusion if there's a reverse jamming phase transition. There are all sorts of other phase transitions in biophysics, and these are things where there changes in the state of, it could be changes in the state of the cell but in what they might be talking about is changes in the state of a cell population. So the abstract reads, to maintain homeostasis, living cells process information with networks of interacting molecules. Traditional models for cellular information processing have focused on networks of chemical reactions between molecules. So this is where the cells are producing um, you know some sort of diffusible substance or secreting some sort of protein, or there's something that they're using to communicate with, like peptides to communicate with other cells. And so that's kind of what we think about when we think about information processing. you know Taking those signals and decoding them, like we talked about with in the last paper with uh, chemical and hardness cues for you know uh, determining which direction to move, or the taxis. So that's kind of what we've been limited to. But what they're proposing here is that the networks of physical interactions might also contribute to the processing of information inside cells. So in particular, we focus on the impact of biomolecular condensation, a structural phase transition found inside cells. So we've talked about cell condensates before in the group. This is where you have different condensates that occur within, I think, within the cytoplasm, and it's this phase transition in different aspects of cellular growth. So they'll focus on biomolecular condensation as a way to sort of get this idea that phase transitions that contribute to this process allow for information to be processed. Biomolecular condensation has recently been implicated in diverse cellular processes. Some of these are essentially computational, including classification and control tasks. We place these findings in the broader context of physical computing, an emerging framework for describing how the native dynamics of nonlinear physical systems can be leveraged to perform complex computations. The synthesis of these ideas raises questions about expressivity and learning. So, expressivity, of course, we talk about that a lot in machine learning and in graph neural networks, and that is where the range of problems that cellular phase transitions might be able to solve is either greater or lessened by the level of expressivity. So, in other words, if I have a combination of things, uh, how how much can that combination of things lead to new things, or how much can that combination of things be informative? So you can have algorithms that are highly expressive or less expressive, meaning that they can solve different types of problems. And in this case, you basically have, with higher degrees of expressivity, a larger range of problems that you can solve with cellular phase transition. So, if you think about a system with low expressivity, a phase transition might only allow you to solve maybe like differentiation of a cell. With high expressivity, it might allow you to do that, plus something at the level of the cell population or maybe the tissue level. And so this is where, it's kind of an interesting way to phrase, um, you know, a way to, information processing as a problem, not only a problem to be solved, but sort of how many problems can this phenomena potentially solve. And then learning, of course, is how these systems could adapt and evolve to solve different problems. So this is basically, how can the cell learn, you know, what to solve and how to adapt to a certain situation. This emerging area of research presents diverse opportunities across molecular biophysics, soft matter, and physical computing. So this is where they get into talking about how living systems are often far from thermodynamic equilibrium. A lot of mechanical systems exist in this thermodynamic equilibrium, but also maybe like a structural equilibrium as well. And living systems don't necessarily do that. And so, we don't really have a good way to model far from equilibrium systems. We usually think of modeling as an exercise in placing things in equilibrium and then testing them. Same thing with a lot of experimental physics, where you assume equilibrium and then you deviate from equilibrium in some way. And you might see how it recovers, but it always has this equilibrium point that you can fix the equation to zero. And of course, if something is behaving far from equilibrium, it's often going to be away from that 0 point. And so it may not recover for a long time, or it may not recover at all, really. And if we think about homeostasis, that's largely an assumption of normality, right? So homeostasis, or a set point, is a set point where the system is operating under normal conditions and it's healthy, and that's how we kind of define a healthy state. But there's no reason to think that that's, that the set point is either healthy or is something that the system operates around. So this is kind of talking about kind of our assumptions about homeostasis and about how energetics is efficient by sort of queuing to this homeostatic standard. And so they go on, even an individual cell can sense and compute to mount an appropriate response to external stimuli or internal changes of state, shown schematically. They were going to show this in figure 1. And then they talk about different signals, environmental cues, and internal signals. And these can be of many different types. And so, we have nutrients and stress coming in from the environment, and then stochastic expression of genes and the depletion of metabolites coming from internally. And so then from an information perspective, homeostasis is a control problem. So they're going to apply a closed loop control system to this. And so again, this is like a model, right? I mean, it's going to describe the system in a way that they're hypothesizing it operates. So, this is, again, just this way, so they kind of point out here, in some cases, cells respond to a relatively simple one-dimensional stimuli, such as the concentration of a single chemical species. For example, E. coli and neutrophils move themselves up a concentration gradient in a process called chemo-taxis. And so this is where these different types of, you have E. coli, which is a single cell organism, and neutrophils, which are in a nervous system. They move themselves according to these chemotaxis. More generally, cells possess a vast array of physical, e.g. matrix stiffness, and chemical signals, e.g. soluble and matrix-bound growth factors. So you can have, not just chemotaxis, but you can have a variety of these different taxis working or semi-independently. There's some joint distribution of them, but mainly, they're different processes that the system is responding to differently, and it adds to this heterogeneity. Processing of these high-dimensional stimuli can include classification, the assignment of a label to a broad range of input values. Classifications allow cells to mount distinct responses to complex stimuli. Indeed, classification has been recognized as an essential step in stem cell differentiation. The patterns triggering particular differentiation pathways require discrimination between many ligand combinations and temporal patterns. So cells need to be able to classify things. And the classification to be correct, it can't have a lot of false positives. And this is important in a lot of processes that are sort of high tolerance or low tolerance processes, where you have to have high precision and you can't have a lot of error, although you can have error in some processes, and there's sort of an error built in, but error correction mechanisms built in. But this is a little bit beyond this paper. In this perspective, we discuss how two emerging topics in physics, biomolecular condensates and physical computing, could complement each other and provide fresh insights and new tools for information processing, biomolecular networks, particularly those inside of cells. And so this is where they talk about biomolecular condensates. So biological cells comprise thousands of different molecular components, which are organized into many spatial compartments. And so while some of these compartments are enclosed by lipid membranes like the nucleus, biomolecular condensates are these domains that are sort of enriched in proteins and nucleic acids. So, you have these like basically clumps of material that have information in them. They're not enclosed like we see in a lot of organelles or in the nucleus, but they're there floating around in the cytoplasm. This figure 1, where we have A, which is where they highlight that the stimuli are multidimensional. So you have nutrients, growth factors, temperature, and elastic moduli. Those are all inputs to the cell, and they're all stimulating the cell, sometimes in different directions. So you have these stimuli coming in, and then you have this response, differentiation, division, metabolism. And so these inputs lead to these outputs, these responses. And the idea is that it's processing this information to get to this response. B is where we have some detail. This is a condensate here, just these green blobs, which are these balls or these masses of different, proteins and nucleic acids and things like that. And so it's, they're enriched with these things and they float around and they're sort of these storehouses of these materials. And it's very important in a lot of developmental processes. So you have this, this is sort of internal to this. So this is information processing between input and output. Okay, so that's figure 1. Then figure 2 is where we have these phase diagrams of biomolecular condensates. So we have 2 different concentrations of condensates, the low concentration and the high concentration case. And you can see in the low concentration case, you have these small globs of things, or maybe they're independent proteins and nucleic acids floating around in the cytoplasm. And then high concentrations lead to this clumping of these different biomolecules together into these large clusters that you can see in the green. So you have to have high concentrations to have these large condensates. They form sort of as maybe a phase transition of concentration. So when you have low degrees of concentration, things are independent. And then when you hit a certain threshold of these things in the cytoplasm, then you get these clusters. And it's spatially heterogeneous. So you might get clusters in one region, but not another. But anyways, this phase transition allows you to have, or allows the cell to have these condensates available. So this is system concentration here, this diagram above these 2 figures. There's this tie line, and then there's this sort of phase transition from individual biomolecules to these condensates. And there's this sharp transition that occurs. That's the canonical system. Then the grand canonical system is where we have some attractive surface forming a subcompartment. So these, this is where this is free floating stuff in the nucleus and it forms this biocondensate. This is in B where they're attaching to a substrate and, again, they're just kind of attaching at random and there's this sort of scattering of these biomolecules on the substrate. Whereas in high concentration conditions, the entire surface of the subcompartment is packed with these biomolecules and their biomolecules competing to get down to the bottom. And, there's this phase transition that occurs. You get this surface that's filled with biomolecules versus the surface that's sporadically covered with biomolecules. And again, up in this figure here, you have where you have a reservoir concentration of biomolecules. As that increases, then you get a switch from phase A, which is this sporadic case, to phase B, which is the packed case. So again, you have these different behaviors, and it leads to maybe differences in information processing because you have different modes of these biomolecules available. So yeah, the composition in the subcompartment in B undergoes a discontinuous jump when the concentration is increased across the phase boundary. Okay, so we're modeling the concentration of this. We're looking at the behavior. We're noticing this phase transition, which can be described by nonlinear physics models, especially this is kind of a, this isn't the formal model, but this is a diagram showing the power law distribution of this. Okay, so typical biomolecular condensates include many distinct compartments or components. Stress granules are thought to have hundreds of components. These components are enriched selectively based on physical interactions, which are dictated by specific sequences and chemical modifications. While there have been no systematic census of condensates, merging the results of independent studies suggests that there are at least tens of distinct coexisting condensates. The compositional variation across different types of biomolecular condensate yields compartments with distinct physical and chemical properties. They can localize biochemistry through enrichment or depletion of enzymes and diverse small molecules, including ions and metabolites. And so macromolecules could adopt novel conformations in condensates that could impact their reactivity. So being in a condensate allows you to have not only a higher concentration and more sort of strength when there's a site of action available, but then there's also this increased reactivity because you can have these novel conformational states that then map to function. Physically, condensates have diverse rheological properties like viscosity and elasticity, interfacial tensions, and electrostatic potential. And then how do you get these phase transitions? So condensation of these biological molecules can be conceptualized as a phase separation, where a homogeneous mixture sorts itself into at least two different macroscopic domains. For systems near equilibrium, phase separation is understood as a competition of translational entropy favoring a homogeneous mixed state and cohesive interactions favoring segregation into different phases. Okay, and so while we consider an entire cell on timescales where production and degradation are negligible, the total amount of material in the system is conserved. And then, we work in the canonical example, or, we have this canonical ensemble, which is where you have multiple phases and they can differ in concentration by multiple orders of magnitude. Okay, so then this second figure, this third figure, figure 3, shows the phase boundaries of multi-compartment systems as decision boundaries. So this is where we have so A is this space of images, and this is just a conceptual model of how decision boundaries work. So like you have the space of images that are inputs to this neural network, and the neural network has to decide whether a certain image belongs in the category of cats or dogs. And so, this is where you have these black points, and each of these points is some variation on cats and variation on dogs. And so, it has to classify them as either a cat or a dog, given the feature space of the images. And so it can do this through the synaptic weights and the synaptic weight matrix Wij. So it's through the connectivity of the system that it's allowing it to process the information so that it can make a determination. So the idea here is that you have some molecular network where you have a bunch of information that's being transferred, and it's giving information about the different features of a biomolecular condensate or some conformational state, and it's trying to make a decision that will put it in one category or another. So the output will be one category or another. The input is all this information, and hopefully the thing does it correctly in order for the cell to survive and to return to equilibrium. Now in a molecular soup, this is not a neural network. They do have the synaptic weights, which is Wij. So they use that in this model of the molecular soup, which is a network of, I guess, uh, we have these different molecular actors and their interactions between them. And you have these binding affinities, basically, which is also WIJs. The idea is that these biomolecules will bind according to affinities WIJ, and they act very much like a set of synaptic weights in a neural network, which aren't actually synapses, but that's the way they refer to them. And then this gives the model another sort of set of information, which allows it to sort of make a decision whether it should switch from context 1 to context 2. So it has this ensemble of information available to it. And from that ensemble, much like the feature space of these images, it can go from one sort of decision-making context to another. And it can switch across this phase boundary when there's a phase transition. So if we think back to our molecular condensates, when we don't have our condensates, when the biomolecules are scattered all over the place, then you're in context 1. When that ensemble changes due to there being a biomolecular condensate available, then the output behavior changes the context 2. So drawing from this analogy, they're able to kind of make this case, or at least partially make this case, for decision making in the cell being driven by these phase transitions. And so comparing figures 3a and 3c, we reinterpret phase transitions in a multi-component system as classification of high-dimensional chemical compositions. Intriguingly, mean field models of multi-component phase separation are analogous to artificial neural networks. They are built upon a network representation of molecular interactions where a set of parameters, Wij, describe the physical coupling of different molecular species, shown in figure 3D. This is here. Just as Wij adjusts decision boundaries as in an artificial neural network, Wij moves phase boundaries in a multi-component system. And so then they talk about closed loop control and condensation. They talk about control systems using feedback and how that's very important. And they talk about a chemostat as an example of this closed loop feedback. So where a chemostat can maintain a fixed pH value within a reactor. Even as non-stationary chemical reactions consume and produce pH-active species. And so phase separation can greatly simplify control into a single physical process without separate entities to sense, compute, and actuate. And so phase coexistence can suppress concentration fluctuations, and that's in this paper number 28. And so that is this paper here in Nature Reviews Molecular Cell Biology 2017. So they kind of go through a little bit more about temporal information processing and how phase separation can kind of model a lot of this and provide a model for these different types of decision making. Okay, so I think that's enough about that paper. I think it's a nice hypothesis-driven paper where they kind of have this idea that this is what's happening in a cell, and there's this whole field of cellular decision making where you have all these inputs that lead to these kind of outputs that are discrete decisions that the cell can make, and there's this effect on collective behavior of tissues and things like that. But it's a nice way to kind of incorporate biomolecular condensates into that.
I like that paper better than the previous one. It seems more real, more realistic.
Yeah.
Anyway.
All right. Yeah. Well, anyways, yeah. So next week maybe we'll do like a CompuCell 3D paper. I'll have to go through and see if I can find something good.
See if they have a one on concentrates.
Yeah.
Or one that's similar to the spots and stripes one. And people have done that for quite a while, making chemical spots and stripes. You could ask Dick about it.
Yeah. Oh yeah, sure. Yeah, that's easy. That's an easy trick. The question is like, how do you attach that to the rest of the biology? Like,
Yeah, well, you've got BZ reactions that do something similar with spots and stripes, sort of a controlled pattern.
right. All right. Well, that's all for today. Thanks for joining.
Okay. Well,
See you next
thank you.
Thanks.